A microbiology study guide for exam 1, Neisseria and Moraxella. General characteristics. What shape and gram stain are Neisseria and Moraxella? They are gram-negative diplococci. This means they have flattened sides. They look like kidney beans or coffee beans with the exception of Neisseria elongata, which is rod-shaped. Are Neisseria and Moraxella oxidase positive or negative? They are all oxidase positive. So all Neisseria and Moraxella are oxidase positive, and that means they turn the test purple within 30 seconds. Do Neisseria grow on McConkey agar? No, and this is actually kind of surprising because they are gram negative, but they are very fastidious and require enriched carbon dioxide conditions, and chocolate agar is really the best. Neisseria is fastidious and need chocolate agar. What are their growth patterns on BAP, chalk, and McConkey? Blood agar, chocolate, McConkey. Variable growth on blood agar, yes on chocolate, no on McConkey. Are Neisseria aerobic or anaerobic? So they're actually aerobic, but they are considered capnophilic, which means they prefer increased carbon dioxide and humidity. Are Neisseria catalase positive or negative? They are all catalase positive. They are also non-modal and non-spore forming. That's implied. So they're oxidase positive and catalase positive, gram-negative diplococci. What is their colony morphology? So when we grow it out on plate, we're going to see small tan colonies. I think they're kind of like pinpoint colonies. Are Neisseria DNA ACE positive or negative? Neisseria are DNA ACE negative. I think, hold on, Moraxella might be, which one? One of them has DNA ACE in them. I think it might be Moraxella. Let me see if I can pull this up. Okay, yeah. So I was right. DNA ACE positive is the Moraxella species, particularly the clinically significant Moraxella catarrhalis, and are generally DNA ACE positive. And it's the key biochemical feature used to differentiate Moraxella from similar looking organisms such as Neisseria. Cool. Oxidase positive, catalase positive, DNA ACE positive. Okay. Which genus are in the family Neisseria? Neisseria echinella kingella. Which genus are in the family Moraxellaceae? Moraxella Acinetobacter. Which Neisseria are primary pathogens? Neisseria gonorrhea and Neisseria meningitidis. Which one out of those two is always a pathogen? Neisseria gonorrhea. Which Neisseria is part of normal flora but can cause disease? Moraxella catarrhalis is it can cause pneumonia, sinusitis, and otis media. So this is one of those other ones with like strep pneumo and Moraxella, and I think you said Haemophilus are the three that can always cause upper respiratory issues like pneumonia, sinusitis, and ear infections. What infections does Neisseria gonorrhea cause? Genital, pharyngeal, rectal, and skin infections. Now, the disseminated infections can cause arthritis, bacteremia, and neonatal eye infections. So remember, it is a big cause of juvenile arthritis. Neisseria gonorrhea is, and then the neonatal eye infections, they use um, eye drops to as prophylaxis for newborns. Are men usually symptomatic with gonorrhea, and what are the symptoms? Yes, urethritis with purulent discharge may spread to prostate or epididymis. Are women usually symptomatic with gonorrhea, and if untreated, what does it lead to? So, no, generally women, about 50% are asymptomatic, and if left untreated, it leads to pelvic inflammatory disease, infertility, endometriosis, and ectopic pregnancy. How is gonorrhea diagnosed in men? Intracellular gram-negative diplococci in PMNs on gram stain, and PMNs are basically white blood cells. In women, the gram stain is unreliable. The culture or PCR are needed. What swabs are used for Neisseria gonorrhea? Dacron or rayon swabs. We cannot use cotton swabs. How should gonorrhea specimens be handled? So remember, these organisms are fastidious. So we can't refrigerate. We need to plate it immediately and transport it to the lab fast. Organisms are sensitive to drying and temperature change. What is the number one cause of septic arthritis in teens and young adults? I actually talked about this a little bit earlier. It is disseminated Neisseria gonorrhea. Blood cultures are often negative due to SPS anticoagulant inhibition. So we're not going to see the growth of Neisseria gonorrhea in the blood culture because the anticoagulant additive SPS will inhibit their growth. What infection can newborns get from Neisseria gonorrhea? So we talked about this already too. Ophthalmia neonatorum, and that's prevented by the erythromycin eye drops at birth. What media is used for Neisseria gonorrhea in sterile sites like cerebral spinal fluid, CSF? Blood agar and chocolate. What media is used for Neisseria gonorrhea when normal flora are present? So the sterile sites were blood agar and chocolate, and when normal flora are present, so not coming from a sterile site, we want to use selective chocolate like MTM. And that's, I think, modified Thayer medium. So what are the antibiotics present in MTM? And that's kind of a connection I'm making right now, is that MTM is actually just a selective chocolate media. So they've added the antibiotics to chocolate. I thought they were two different ones. And if you can remember, try and tell me what each antibiotic kills. So vancomycin kills gram positive Colistin kills gram-negative. Nystatin kills fungi. And trimethoprim kills swarming Proteus. Vancomycin, what does it kill? Gram-positive. Colistin, gram-negative. Nystatin, fungi. Trimethoprim, Proteus. Proteus. Okay. Vancomycin, colistin, nystatin, trimethoprim. We do it. Trying to think of a way to remember that. Vancomycin, colistin, nystatin, trimethoprim. Vancomycin, colistin, nystatin, trimethoprim. Vancomycin, colistin, nystatin, trimethoprim. Okay. What do Neisseria gonorrhea colonies look like on chocolate? Small, smooth, glistening colonies. What molecular test is used for gonorrhea? PCR or a nucleic acid probe. They can detect Neisseria gonorrhea and trachomatis together, so co-infection up to 41%. Oh, that's chlamydia. Gonorrhea and chlamydia are tested together. We're looking at Neisseria meningitidis now, so this is another big one. What are the major virulence factors of Neisseria meningitidis? It has pili and a capsule. So remember that picture from the video where it has the little like spacesuit capsule and pili inside. What disease does it cause that is highly fatal? Meningitis. So even if treated, mortality can be 25%. Where do outbreaks of meningococcal disease happen? Dorms, barracks, and prisons. What is meningococcemia? Sepsis with or without meningitis, and it can cause Waterhouse-Friderichsen syndrome, which once you get there, you can die pretty quickly. You've got petechial rash, shock, organ failure, and rapid death. So the Waterhouse-Friderichsen syndrome is associated with meningococcemia, the sepsis with or without the meningitis due to Neisseria meningitidis. How should CSF specimens for meningitis be handled? They are a stat test. They need to be transported quickly. Do not refrigerate. And cytocentrifugation improves detection. Which Neisseria is normal flora in the nasopharynx? Neisseria lactymica. Which Neisseria is rod-shaped? Neisseria elongata. What infections does Moraxella cause? Pneumonia, sinusitis, and otis media. Remember, this is one of the three that can cause all that infection in your upper respiratory and face and ear area, along with strep pneumonia and Haemophilus. Is Moraxella acircolytic or saccharolytic? It's actually acircolytic. It does not ferment any sugars, the glucose, maltose, lactose, or sucrose. Is Moraxella DNA ACE positive or negative? It is positive, so it's one of the things that really separates it from Neisseria, because Neisseria is DNA ACE negative. What is the colony morphology of Moraxella on blood agar? Medium white convex colonies, and the big thing is that it moves like a hockey puck when pushed. Hockey puck colonies for Moraxella. What does Moraxella look like on a gram stain? Now remember, it's part of this little gram-negative diplococci family, but essentially we're saying large gram-negative diplococci. What does Moraxella grow on MTM? That's the modified Thayer medium. Remember, that is the selective chocolate with all the added antibiotics like vancomycin, nystatin, and tropomycin. What's this like? Colistin. It's inhibited on MTM, but it grows on blood agar and chocolate, regular chocolate. What sugar does Neisseria gonorrhea ferment? Glucose only. Which sugar does Neisseria meningitidis ferment? Glucose and maltose. Which sugars does Neisseria lactamica ferment? All three, glucose, maltose, and lactose. Which sugars does Moraxella ferment? None. And that's all.